And we will move to our next speaker, which is from the same lab as Javier briefly also introduced, is Cecilia Herbert. She's a PhD student in the lab of Gabriel Mindlin at the Institute of Physics in Buenos Aires. And she will tell us now about actually these coding schemes, I think, that Javier introduced that could be studied in the canary song. So stage is yours, Cecilia.
Thank you. Thank you for that nice presentation. So can you see my presentation? Fine. Great.
Yeah,
so yeah, I'm going to continue to tell you about the work that Javier just described. Given that behavior in canaries is better suited to test the gesture transition hypothesis, I set out to test the predictions of a neural model also developed in our lab using electrophysiology. And this is work I did with Santiago Boari and Gabriel Mindlin and Amador. So here's an example of canary song. This is a spectrogram of the song. So that's a brief snippet of what the animals sing during the day. And you might see the long whistle syllables that Javi just described and the short syllables here. And these are the repetitions of syllables which make up phrases that make up the song. So this is another animal as the spectrogram and the associated air sac pressure required to produce these syllables. So the long whistles are typically produced with this pressure pattern called a P0 pattern, and other short syllables have this other pattern, P1 pattern. So this pulsatile expiratory gestures. And this is work, this categorization was done by our lab, and it will help us to group these syllables together. That's why I'm showing it to you now. The other element of this is the Oscine song system. We're looking at HBC, but this is a system in which the motor pathway is shown here in black and in green. And the neural model I'm telling you about was developed in our lab. It follows this circular architecture, so it's called the circular model, and it joins HBC with RA with the expiratory areas in charge of generating these pressure pulses that I mentioned, and an initiating area that gives rise to the instructions going to this circuit. This is a rate model, so we're going to be looking at summation of activity of individual models as activity over time. And the model was able to reproduce experimental air sac pressures that I showed you just now. So this is a robust neural model that produces the behavior that we're looking at. And I'm going to tell you about the predictions in HVC in a little while. But to test this model, I had to develop and record electrophysiologically in nucleus HVC. And this is the device that we used. It's just a micro drive driving tetrodes into the brain, so micro wire tetrodes. And we constructed it in-house since we had to make it ultra lightweight for implanting it on the bird skull and compact. And using tetrodes also fabricated in-house made that it was customizable and low cost. So here's a bottom view of the tetrodes. And this allowed us to perform chronic neural recordings for around 7 weeks in most animals. So with this device, we recorded activity during song. Here you can see for a P0 phrase, the example sound pressure, the spectrogram associated with the song with the long whistles, and example activity for one neuron, an isolated neuron firing bursts correlated with behavior. These bursts are firing locked to syllabic rate, so once per syllable. And you can see this is the case in P1 syllables as well. So a different neuron, a different animal firing locked to syllabic rate in another kind of phrase. So these basic neurons are the ones that we grouped activity across birds. And I'm going to be comparing that to the predictions of the circular model, which for P0 syllables involve an initiating area having 2 kicks of activity and two sparse pulses in HVC during the start of the syllable. If you notice, this is the pressure pattern, remember, for the P0 syllables, and this is the long whistle part. And there we don't have an associated activity in HVC. So this reminds of the gesture transition model that Javier was, hypothesis that Javier was telling you about. So no bursts in HVC here, predictive. And this is the recorded in activity in HVC during song in 7 neurons for 5 different animals. And this is a raster plot of the activity of single neurons. And here you can see an example pressure pulse. So the summation of this activity is shown here in green. And you can see that there are pulses of activity in HVC at the start of the syllable, as I was saying. We couldn't find for these amounts of neurons and bursts any firing during the whistle. And there we did find something that was not in our model, which could help future iterations of the model, which was some sort of activity for the termination of the syllable.
Cecilia, sorry to interrupt, but we need to wrap up. Thank you.
Wrap up, perfect. Yeah, so we found the same this to be in P1 phrases, and in conclusion, our experimental recordings support the circular model. So thank you for listening.
All right, thank you very much. Very interesting results. So I think we have our first question, and the question is from Conrad, and he's asking, well, I don't know if Conrad wants to come live and ask the question. I think so, right, Ishaant? Okay, otherwise I will read the question out. Okay. So the question is, in a lot of mammalian cortical cells, there is firing to many other things. Are these neurons totally silent when the bird does not sing? Or are they really specifically tuned to bird songs and not doing anything else?
During production, they're actually hard to pick up, like these projecting neurons fire very sparsely, and you might have seen that I don't have tons of neurons, and I did tons of recordings. So they're hard to pick out, and they do start firing phasically during specific phases of the behavior of the bird's production of song. We're not recording them outside of any other behavior. There is HVC does serve as a hub for other kinds of activity, but as far as we know, like they are starting to fire basically locked to song production. And they do, as Javier said, in some animals they do, in some species they do have this mirror activity, and they fire while the bird is listening to their own song. And in canaries, we're looking into that at the moment. So maybe next conference.
Actually, I mean, related to what you just mentioned, that they are quite sparse, and it's technically also hard to record from these neurons. Do you try, for example, as you said, because you know they're responsive to the playback of their own song as well? Do you try such tricks to be able actually to increase your yield of recordings, like playback their songs, so maybe it would be more active, so you could have more neurons?
Well,
let's say, yeah.
Yeah, no, this one is like an unbiased recording. Like we're just stimulating the animal to sing in many ways, doing some playback, but just like to get them to sing, not necessarily looking for a response. And in canaries, it's still not, I mean, we're looking at the moment, it doesn't seem to be the case that the same neurons that respond to the auditory stimuli respond to the I mean, are active during song production. So we're trying to figure that out at the moment. Some of the neurons I found do have that, but others don't. So yeah, it's not as reliable as a mechanism for fishing neurons. I mean, I think the way to go there would be some sort of imaging, you know, to get some a wider field of view, but then you lose the time resolution. So yeah, it's hard.
Thank you. And or neuropixels.
Well, yeah, I mean, if, yeah, of course.
Yeah, promoting now.
But I wouldn't, I mean, I don't know, would you implant chronically for 7 weeks with a neuropixel?
Well, I think there are actually, I think even in this conference, we are going to hear about some people that have implanted and they're recording chronically with neuropixels probes as well. Nice. Yeah, and yeah. Which, yeah.
We so don't have the resources at the moment to carry out.
Right. Okay, okay. And we have the next question from Kelvin Jones, and he's asking if it is possible to apply local cooling in the circuit and test predictions of your circular model.
Yes, it seems that that you have been reading the literature from the lab. I mean, it is true that in HPC that we have done some local cooling, and we see we see the predictions of the circular model testing there as well. Yeah.
Okay. So you have done this experiments?
Yeah, no, I mean, not my experiments, that they were done previous, and I think they were during auditory, not production, but yeah, that has been.
And then when you were speaking about this projection, you call them projection neurons. Is it, so what was, because I'm not very familiar with the field, is it like, so you kind of dissociate between the projection targets, like whether they are innervating different downstream areas, or just as, so somehow you're labeling these cells, or no, you're just recording from every neuron there?
No, technically that would be brilliant to actually do some antidromic stimulation to figure out who they are. Until now, we characterize them based on the electrophysiological behavior. So projection neurons in these nuclei, in HVC rather, are these phasic firing ones. So that's the characterization. So the results I showed were of phasic firing neurons, and those are putative projection neurons, exactly. Yeah.
Okay, okay. So and again, I'm gonna make a kind of a loop to the first question. And since normally, like in the mammalian cortex, when we have these premotor areas, or at least speaking of cortex, sometimes they are responsible for different, let's say, motor outputs, not necessarily one body part. Like in this case, okay, we're talking about complex motor behavior, Birdsong generation. But is there any studies or anything that suggests also the implication of these neurons in other motor control or any other type of movements of the birds or so on?
Not as far as I know, but it is hard to it's like which population of neurons, like it's just a general thing at the moment, right? The whole like projection neurons, or so maybe there's something comes up if we can get to see like different subcircuits within HPC that are currently not described. I don't know.
Right. For example, in the rodent field, there's I would say a tendency to like more and more sophisticated ways, try like high-speed filming and so on and so forth, try to really quantify different behaviors, like even like the small, very like small movements of their facial movements or like forelimbs and hind limbs and so on. And we see a lot of correlations of those type of movements during like task performance and so on in a lot of these premotor cortices. So I was wondering if one has also tried to really quantify such behaviors and see if there's any correlation between the activity of the HPC and such behaviors. But yeah. Very interesting. Thank you very much, Cecilia. I think now we can move to our next speaker. So we're going to move from the songbird. Before that, again, I'm going to ask for a round of applause for Cecilia for a great talk and answering the questions. Thank you very much.
Thank you for having me.
Thank you. And so with that, I'm going to